Good morning, friends. Welcome back to our paper CHO 5403 Biomolecules, Organic Chemistry of Drug Design and Chiral Drugs. Friends, in this paper, we are discussing about the amino acids, proteins, enzymes, and lipids. So, we have started with the introduction of amino acids. Hope you understood the definition and the drawing of amino acids. Friends, also we discuss few important method of amino acid synthesis like Strucker synthesis, modified Gabriel synthesis, Erlenmeyer synthesis, and so on. So, in today's lecture, we will focus on the peptide. So, what are the peptides? Peptides are the short string of amino acids typically consist of 2 to 50 amino acids. The important reaction of amino acid is the formation of peptide bond. Peptide bond is nothing but a simple amide bond between two amino acids. So, amides are the most stable derivative of amino acids because you know that there is a partial overlap of double bond on the carbon and nitrogen. So this restricted rotation of carbon nitrogen double bond, there is a stability to the amide derivatives. So amides are the most stable derivatives of amines and acids. So friends, this amide bond is also called as peptide bond. Okay, this amide bond is also called as a peptide bond. It is nothing but a bond between carbon and nitrogen. Let's see in the next slide. So friends, the amide linkage as we discussed, amide linkage is known as peptide bond. The product is an amide called dipeptide when it consists of two amino acids. That means if we have these two amino acid and if they are forming peptide bond, the product is called as dipeptide. So once we form the dipeptide or tripeptide or polypeptide, each amino acid unit in this peptide is called as residue. That means glycine residue, leucine residue, isoleucine residue, and so on. So in this manner, any number of amino acids can be bound together to form a continuous chain. This continuous chain may be called as a polypeptide, but this polypeptide have a molecular weight less than 5000. So friends, if we have the polypeptide chain which having the molecular weight more than 6000, it is called as a protein. So polypeptides are the next smallest version of proteins. So this is how peptides and polypeptides are formed. So this is a bradykinin, a human hormone which is shown in here. Okay, I am not talking about this hormone, but I am talking about the polypeptide chain. So friends, how we should write the polypeptide chain? The end of the peptide with the free amino group should be written at the left, and it is called as N terminal end or N terminus. Whereas the end with the free carboxylic group, that is this COH group, is called as C terminal end or C terminus. So friends, we must write down the amino acid in such a fashion that N terminal should be on the left side, whereas the C terminal should be on the right side. So how to nomenclature the peptides? So very interesting method is for nomenclature of the peptide. The names of the peptide reflect the names of the amino acid residue involved in this peptide or amide linkage beginning at the N terminus. So N terminus is at the left side. So we will start with the N terminus, and the last part we will have the C terminus. So friends, to nomenclature any peptide, we will start from the simplest dipeptide. So if we have alanine and serine, two amino acid forming a peptide bond, we can use the YL suffix because of the acylation of this amino acid. So the first will have YL suffix, but the last or C terminus amino acid will be written as it is. So what will be the name of this dipeptide? It will be alanyl serine. Okay, this will be alanyl serine. As this is the last residue, so we will not use YL suffix for this, but before that any amino acid is there, we use YL suffix. Okay, I have another example for you guys. This is a bradykinin, a human hormone. So how this bradykinin is named in general? So you know that N terminus amino acid is arginine, then proline, glycine, phenylalanine, serine, proline, phenylalanine, and arginine. So how we can nomenclature this peptide? So we need to add YL suffix to each and every amino acid except the last one. Okay, last one is C terminus. We will not use YL suffix for it. So name of this bradykinin is arginyl, propyl, propyl, glycyl, phenylalanine, ceryl, propyl, phenylalanine, and finally arginine. So this is how we are going to nomenclature the peptide, or simply there is a method to write alaser. This is a short form of this peptide in the 3 letter short form. Okay, so alaser means alanine is at the N terminus and serine is at C terminus. So you can write like this also and this also. This is how we are going to nomenclature the peptide, and we will see how they help to form the protein in the next lecture. So friends, if you have any doubt regarding this lecture, please feel free to ask me. Thank you.